We've got a really exciting discussion planned for today around clinical trial efficiency using data and digital health technologies. What I'm really excited about this discussion and what we'll do during our time together today is we'll go beyond endpoints to think about how can we make the most of the new technologies and capabilities that are at our disposal with the advances in digital medicine more broadly, and think through how we can create value by maximizing operational efficiencies in our trials. It's something that we've heard a lot about from several of our partners here at Dime, is that operations are the hardest part. And so I love that this webinar will be thinking through how can we streamline operations using these new tools in the toolbox, almost inverting that question on its head. Each of the three speakers that we have here today, Michelle Holko, who is Vice President of Biorisk at Airfinity, Abigail Levine at Evidation Health, who serves as a Senior Director of Product and Portfolio Strategy, and Jacek Urbanek, who is a Director of Biostatistics at Regeneron. Each of these folks will be walking through a few exciting case examples where they've been able to drive efficiency through new digital capabilities and share operational lessons learned, so that as we continue to pave new ground, we'll also get better and better as we do so. Before we dive in here with the case studies, please note that today's session is being recorded. The slides and the recordings will be available on Dime's webpage after this session. Just ask that you do raise your hand if you have any questions. We're happy to unmute you and ask you to ask your question live, or if it's easier, please feel free to type your questions in the chat. Sound good? All right. With that, I'm going to go ahead and rather than me introducing each of our webinars individually, I'll ask each of them to share a little bit more about kind of who they are, what they're doing, and some of the exciting work that they're leading at their respective organizations. So Abby, would you like to start us off here?
Yeah, thank you so much, Sarah. I am so thrilled to be here today. I'm Abby Levine from Evidation Health, and I lead product strategy for our consumer platform. I've been with the company for about 7 years and have over 2 decades of research experience myself. I've worked across our decentralized trials and now operating in our direct-to-consumer line of our business. I have a couple slides that I want to walk through for those of you who may not be familiar with Evidation. We are experts in collecting data directly from individuals and in longitudinal data for everyday life. And what we've called this type of data over the years has changed. First, we talk about wearables and we talk about surveys. Then it was patient-generated health data, or PGHD. And now we talk about this in terms of lived experience data. And this, in our view, it's real-world data, it's real-world evidence, but it's maybe not conventional. And what we are seeing, and really why we're here today to talk about this, is that medicine is, I think, starting to recognize the true value of this data as we bring new products to patients. And that we believe that engaging patients over the long term and collecting this lived experience data before, during, and after trial participation is key to actualizing what really has so far been unrealized benefits to patients. This is across really a myriad of domains, but primarily this is increased personalization that leads to improved outcomes. Sarah, I think we can go to the next slide. So at Evidation, we primarily operate in the phase 4 post-market space, and we do a ton of work to help our partners more deeply understand their audiences using a variety of different data types, data sources. And here on this slide, you've got on the left conventional RWE sources, and contrast that with our direct from patient data on the right. We certainly don't believe that one source of data is sufficient to provide that elusive complete capture or that 360-degree view of the patient experience. And what we have been able to do over the last decade or so is build this privacy-first technology that enables taking a bunch of really heterogeneous data, layering it on top of one another, and making it useful so that analysts, end users can generate novel insights to help drive business decisions, and the like. And the ability to do this, in my view, is really important, because what we've seen, I think specifically over the past 6, 7 years or so, is the wearable data, maybe some of this direct from patient data on its own, isn't really all that useful, all the time. Of course, there's certain instances where that may be the case. But when you can start linking or indexing this direct from patient data to some type of clinical event or measure, all of a sudden you've unlocked so much utility. The relevance of these measures dramatically improves. And this lived experience data can also help to contextualize the factors that influence outcomes. It can provide an objective lens into a patient's quality of life. And we're talking about things that you don't get if you're just looking at data from, say, periodic health system interactions or in-clinic assessments. And a lot of this too can relate to how the patient views their own quality of life. What are the things that they want to do? What are they able to do, say, before or after an intervention, or that aligns with, say, some of these more traditional measures. And as I mentioned, what we've learned is that digital data is really, as Sarah mentioned earlier, it's more than just about endpoints. It's really about fully and deeply understanding the patient experience of a disease or a condition. And I believe that it's really going to be some of the hardest to treat, or really hardest to categorize and characterize, chronic conditions where we're going to start seeing some of the greatest potential for impact, some of that greatest impact. Because what we're able to do is start to quantify so many things that may be otherwise invisible. This, as I alluded to a bit earlier, is your symptom burden, the impact on quality of life across n number of domains, minute signs of progression, and other factors that can influence your outcomes. So I want to take you through a couple of case studies, a few research case studies where we've leveraged digital devices and how we've used these to uncover novel perspective on these conditions that lead to just increased operational efficiency, both in trials and in preliminary research. And again, really more holistically understanding your target audience to optimize for your trial design. All right, next slide. So our first example is in women's health, where we've used wearables to measure the impact of endometriosis and uterine fibroids on daily activity. And this is really one of my favorite examples to share, because it really demonstrates the power of some of these digital data streams for understanding chronic conditions. And for endometriosis and uterine fibroids specifically, the importance of this really cannot be overstated. Symptoms are often subjective. Surgery is often required to obtain, an official diagnosis, and this can be a huge confounder, often for real-world evidence or clinical studies, and contributes to significant underrepresentation among your study populations. And I'm not talking just about demographic categories, although obviously that's a huge one, but also with respect to prevalence and outcomes. And so here, what you can see is that we've successfully characterized some of the key differences between diagnosed and undiagnosed individuals that are only visible in this activity data that they contributed via their wearable devices. And so here we've really just started to scratch the surface to start to break down some of these characteristics and really home in on how we can more deeply understand a chronic condition within a population. All right, next slide. Great. Next, I want to highlight our Flu Smart program. So this we run in our consumer app. Our app is available on the App Store. It's really exists for individuals to download and start earning rewards for doing healthy behaviors. They're able to contribute activity data. They're regularly completing surveys, PROs, et cetera. And what we've brought to our members is this remote monitoring program that leverages signals from consumer grade activity trackers. And this includes, Fitbit, Garmin, Apple Watch, to help detect symptoms of influenza-like illnesses. Includes flu A, flu B, COVID-19, RSV, and bringing awareness to flu-like symptoms, especially during that onset period, it's really useful. And I say this because what we've discovered is that people aren't always great at recognizing that they are getting sick until it's too late and they are very unwell. We all know that we have very busy lives. You may feel a little bit run down. How can we start to distinguish maybe just stress in everyday life from something that may require some sort of action on your part? Um, so what we're able to do via Flu Smart is by alerting someone when our machine learning models predict that they might be experiencing symptoms, we can send them a nudge and kind of nudge them to that next best action. And this can be a personalized experience, like maybe you should take a test, maybe you should contact your provider. And in research context, we've actually used Flu Smart and this alert flow to recruit for studies where the study itself required their participants to be within the first 48 hours of the onset of symptoms. And this technology also lends itself to smart diary functionality to improve symptom logging during studies, et cetera. We've seen this particularly for, say, a vaccine trial where it's really critical to document symptoms or the onset of illness as soon as you start to experience it. All right, next slide. All right, and this is our last case study. It's a bit different from our other two, where we were primarily leveraging the data collected via our consumer platform here. This is a study that we ran in cognitive decline, where we combined passively collected data from a number of sources and sensors with data from active tasks and tests, and ultimately extracted over 1,000 features in total. Now, I can certainly relate here, analyzing and contextualizing so many features and generating meaningful insights or conclusions about, say, cognitive decline. It's not really straightforward. There's not a blueprint for this. So what we have done is create what we refer to internally as a behavioragram. And this behavioragram aligns data streams along time series to create the visualization that you see here. It kind of looks like a little bit like an ECG. But what each of these rows represent is a different channel of data. And these relatively dense behaviorograms afford a multi-dimensional view of individual behavior over time. These visualizations help us put data into context and extract additional insights from it. This is really a key first step when looking to analyze such volumes of very dense activity data. For example, we can start to more easily analyze, say, how heart rate changes when people are in a workout session, or the effect of, say, talking on the phone has on walking pace. And what's really effective here is that this approach has allowed us to derive new data channels from the existing ones. So these are just a few of the examples that I wanted to showcase and talk through today. Happy to dive into things a little bit more detail in our Q&A, but for now, Jacek, I will turn it over to you.
Thank you so much. My name is Jacek Urbanek. I'm a director of biostatistics at Regeneron Pharmaceuticals, and I run biostatistical engineering working group where we work with all aspects of digital health across all the stages of drug development. Before moving to the industry, I was an academic, and I've been working with digital health since 2013, so quite a long time. Today, I have one specific case study. It's quite scientific because of the time constraints. I'm not gonna go to details, but you can always reach out to me with any additional questions. And I know this presentation will be recorded. So today I wanted to show how large-scale observational studies, studies that published free open source data can help with rapid decision making when deciding the digital measurement strategy for clinical trials. I'm going to focus on specifically on Parkinson's disease. Let's go to the next slide. So for those of you who work in Parkinson's disease, you know that there is still a need for more sensitive markers of Parkinson's disease progression. Instruments that we have right now are subjective and very noisy, and therefore they require a large sample sizes to prove statistical significance and quantify change related to disease progression or to the drug effects. So we are all searching for new ways and new measures to assess Parkinson's. There are many existing solutions out there, at least when you Google it. But in reality, there's also a lot of noise. Bringing new solutions, bringing new technologies to the large organization requires evaluation. Evaluation takes time and might require running additional studies. Let's not forget that, digital medicine and digital health technologies are not new to us, but they are new to most of the people. So we really need to build the trust internally. And here, we wanted to explore if wrist accelerometry have potential to monitor Parkinson's disease, and again, try to shed some light on that question with open source free data sets collected as a part of National Health and Nutrition Examination Survey Enhance. I bet many of you heard about this study. Few of you probably work with this data already. Let's go to the next slide. All right, so I'm going to quickly tell you what are wearable accelerometers and what's their potential, and then what is the link to Parkinson's disease. Wearable accelerometers measure movement. That's it. They measure movement of the body part they are attached to. They can be worn on different body parts. Today, we're going to focus on wrist, but people also use lumbar location or ankle location. They monitor continuously as long as they are being worn, 24/7 for days or even weeks at a time. And at its core, if you take accelerometer, open it up, look under the microscope, it's just a ball on the spring. That's what it is. Everything else that comes out of accelerometer is a result of algorithms and estimations. Let's go to the next one. Now, when you collect accelerometry data on one person for long enough, let's say 24 hours, like here on the screen, you can characterize quite a few very interesting things about the way people move and how active they are and how they look like. So here I'm giving examples of potential measures, right? We take the total acceleration, sum it up, we're going to end up with the measure of total daily activity. If we see that the magnitude of acceleration go above the certain threshold, we can quantify time spent active, or even go further and quantify things like fragmentation of physical activity, so probability of breaking an active bout and becoming sedentary. And because we collect this data continuously 24/7, we can also look at diurnal characteristics. So we can take a look only at morning hours to see, for example, if we see effects of morning stiffness. We can look at nocturnal activity and try to quantify characteristics of sleep. Let's go to the next one. Right, using accelerometers in Parkinson's research is not new, and this is what this slide talks about. A number of papers with the keyword wearable accelerometer or accelerometry and Parkinson is growing every year. In general, number of publications with the word accelerometer is growing. So we have a good basis to expect some use of accelerometers in PD. However, what the next slide shows is that number of clinical trials in Parkinson with accelerometry is still relatively small. Number of trials with accelerometers in general is growing, but the Parkinson field is still opening up to the idea. If we go to the next slide. Now, when we started thinking about major concepts that are specific to Parkinson's, like low physical activity, impaired mobility, fatigue, stiffness, decreased sleep quality, increased nocturnal movement, plus tremor, bradykinesia, dyskinesia, all of these things can be measured with accelerometry. You can, when you think about how people move, both throughout the day on the macro scale, as well as on the micro scale, like the tremor, shaking of the wrist or slowness of the movement, wearable accelerometer seems to be a good technology to explore. Let's go to the next slide. Now, to explore it, to show, to gauge if there even is a potential, we turn to ENHANCE study. ENHANCE, it's a cross-sectional study. There are cohorts every 2 years, but the cohorts are independent from each other. So unfortunately, there is no disease progression. It collects a lot of different questionnaire type outcomes. It has limited clinical assessment, and some cohorts have accelerometry measures. Now, we don't have, for most of these people, we don't have diagnosis. So we had to infer if they have Parkinson's or not. But we had information about the medications they are taking. So based on the list of medication, we identify likely Parkinson's patients. We also identify people who might take Parkinson medication for Parkinsonism and rule them out. And for the people we had 7 days of free living accelerometry, we match age, gender, BMI, and ethnicity-based controls. We ended up with 20 Parkinson people, 20 controls, and we calculated few accelerometry based outcomes using open source R package, ArcTools. Look it up. That's the fruit of my collaboration with few other very talented folks in academia. And we started exploring. Let's go to the next slide. So through the exploration, we were able to a good degree, replicate previously published results. So the plot on the left shows diurnal patterns of people taking Parkinson's medication, our likely Parkinson's patients in blue, and our controls in burnt orange. The plot shows 24 hours of average physical activity starting from midnight to midnight. So you can see during the night, people are not very active, and throughout the day, the activity goes up, and Parkinson's people are less active than our controls, Parkinson's free controls. And on the right, the diurnal plot on the right, these are the results published in, I think, Nature Digital Medicine by Schalkamp using UK Biobank data on much larger sample. And we were very pleased to see that there is a strong resemblance. Therefore, we came up with a few hypotheses to test, namely that people with Parkinson will have a lower total physical activity, lower morning physical activity, higher nighttime activity, lower activity intensity, so how intense they move disregarding the volume, and their physical activity will be more fragmented. There are outcome measures already available for all of them, and they are to a large degree well validated. Let's go to the next one. Now, I'm not going to spend too much time on this slide, but we only identified 20 people with Parkinson and 20 controls, but it doesn't mean we discarded the rest of the data. We actually use the rest of the data to calculate reference values to help us contextualize the accelerometry outcome. So basically, we came up with the percentile curves, and this is a background, the broken lines on this plot to show how these accelerometry characteristics distribute across the entire population. If we go to the next slide, what we found out was that indeed people with Parkinson's have lower total activity. They move less vigorously, so they have lower intensity. They are more fragmented, and their morning activity is also lower. Morning activity is very important because Parkinson patients usually take levodopa medication to alleviate their symptoms before going to bed in order to sleep better, to be less stiff in bed. And by the time they wake up, the effects of drug wear out. So the severity of their symptoms is likely to be the strongest in the morning. So we were able to observe that, and we were not able to observe any difference in how they sleep between healthy and controls. But these results were very promising, and I think my next slide is my last slide. So because of that, what we were able to do is we were able to show the potential of technology without running any internal study. We built the trust of our senior leaders in accelerometry. We show that we can handle the analysis of these voluminous data sets. What we haven't shown is, well, can accelerometer capture actual progression of the disease? That is still to be addressed with internal studies. However, it gave us very good foundation to craft a well-informed hypothesis with almost no cost. That's it. Thank you, guys.
Awesome. I will take over from here. Thanks so much, Jacek and Abby. I'm really excited to be here to talk with you all today. So my name is Michelle Holko. I'm the Vice President of BioRisk for Airfinity. Airfinity is a disease forecasting company that's based in the UK and got its start in 2015 as kind of the Bloomberg of life sciences and doing reporting on discoveries that were coming out of the life sciences industry, because as we all know, it takes a long time to get papers published, and that's kind of the primary way that people share their results. And so the vision of this company was really to bring this information forward and to have it be a lot more useful to other companies in the space, as well as just other consumers. And then certainly throughout COVID, the focus shifted hugely to infectious diseases. So really with Airfinity, the core proposition here is around data. So it's different than Jacek and Abby's use cases, which are really focused on digital devices and the data coming off of those devices. At Airfinity, we're solely focused on the data and playing the long game with the data. And what that looks like is infectious diseases data, data around what types of medical countermeasures, whether that's therapeutics or vaccines or other solutions that are in the pipeline, how long till those are going to come to market, what is currently available, how much of these different products are available. And then we also have a much more recent product that we've just launched around obesity and some of the anti-obesity drugs and cardiovascular disease. So, you know, the focus is around data. We scrape data from over 5,000 sources across the web. This includes epidemiology data. It includes clinical trials data. It includes cases, deaths, hospitalizations, but also things like climate data, mobility data, travel data, wastewater data, certainly. And we're also really interested in exploring a partnership with Evidation around how we can integrate data from wearables in the context of this work. And so what do we do with this data? Well, you know, one of the things that you can do with data once you have enough of them is you can start to see the patterns and what they're telling you. And so a lot of what we do is analysis of the data, but also forecasting and model building so that we can say when we think that there is going to be a surge in a specific disease, where we think that's going to happen, and what the timing of that looks like. So again, multiple sources of data, you know, we gather multiple sources of data because there's a lot of data missingness, which I think is one of the issues with devices as well as with these kinds of publicly available data, is that there's often a lot of missingness. And so it makes it really hard if you're reliant on a single source of data to make these determinations. But if you're gathering lots of sources of data, then often what you can do is use data triangulation, or looking at evidence from other sources of data to infer what's actually happening. And so we work with pharma companies, biotechs. We also work with governments across the globe and non-governmental organizations. And the use case that I'd love to talk to you about today is squarely focused on improving clinical trial efficiency by using data around infectious diseases to target the location and the timing of recruitment, so that if you're reliant on a specific disease burden of an infectious disease to test out your vaccine efficacy or your therapeutic efficacy, and you know, you start this trial and then there's no disease circulating, well, that's a lot of wasted effort and money, and it's going to take a lot longer to get through the regulatory process and to see how effective that is. And so what we do is we combine our decades-long data core to understand where these outbreaks are going to happen, what the seasonality and timing is. Another reason why this is, I think, a really important use case is, you know, when it comes to a new infectious disease threat, we all experience this with COVID, the time that it takes from sensing this new pathogen to having a vaccine or a therapeutic available means lives saved. And so time is really of the essence, and you want to be as efficient as possible, and you want to be as quick to market as possible, so that you can start to protect lives. So that's one of the reasons why we think this is a really good use case for this type of approach. Another reason is for diseases that are circulating, so that's for new and emerging threats, but for diseases that are circulating like RSV or flu, there's a seasonality to this circulation. And one of the things that has been noticed is that if you focus on just looking at the attack rate in one season, and you base your clinical trial design on that one season's attack rate, you're missing a lot of the equation. And oftentimes the studies are underpowered and they end up not being significant. But if you look at 3 or more seasons, and you build that type of analysis into your clinical trial design, your statistical power goes way up, and you're able to buffer against those kind of normal fluctuations that we see season to season in infectious diseases. So in terms of some of the benefits, certainly increasing recruitment, using this kind of an approach, decreasing your costs and also accelerating your timelines are some of the major things. It can also, if you're flexible in terms of your strategy, in terms of locations, it can also help you decide which locations to ramp up or ramp down as the trial progresses. So kind of getting to a little bit more to that, precision clinical trials. And then another aspect that we have looked at is how do you know how to recruit the patients that are going to be most useful to your study, that are going to be most impacted, and that's another area that we look at a lot. Especially in the infectious disease area, we look at patients that are immunocompromised, that are specifically in need of these monoclonal antibody solutions, because they're really challenging in terms of mounting an immune response to the vaccine. So there are a lot of different ways that you can go about that. And so I guess one of the things that I'll end with is that I think, as we're all talking about all of these very different use cases, I think one of the things that is the common thread is the data, getting insights from data and planning your study in advance, and also being able to be creative about where you're getting data from. Accelerometers didn't exist in clinical trials a long time ago. And certainly the wearables data is still kind of a new piece of the puzzle. So really excited to see Evidation and others leaning forward in this space, and happy to take questions.
Excellent. Well, we just heard from three amazing leaders across the field, three amazing use cases. And certainly if you do have questions, we'd welcome you to put those in the chat. But as those are coming in, I know I have a couple of questions for you all. I think we heard a little bit about some of the different use cases that we could leverage this data for beyond just endpoints, right? But things like improving recruitment, enrollment, time to actually conduct this study and that type of thing. But one of the things that I wonder, and maybe I've been asked a lot, frankly, is as we continue to gather this sensor and patient-generated health data, how can we ensure that it's not just the quantity of the data that we're looking for, and the different data sources, but how can we ensure that the quality of the data is there as well, to ensure that we are using trustworthy data in order to continue progressing our comprehension of these different disease states.
I could maybe start on that since, our approach is a little different, but actually that's an incredibly relevant question to the work that we do. And I'll say that, at Airfinity, we have about 100 people in the company, and the majority of these people are working on data or analysis or modeling. So most of them are, PhD or master's level experts in their field of specific disease area. And it requires that human in the loop for this use case to understand, this media report that came out, so we also scrape media data, which, that's a very different trusted source than a scientific publication and the data table that comes out of that, or reporting from a public health agency. So those media data, although it's incredibly important to us because we want to be first to notify, we also require people to actually look at that and vet that and say, are we getting that same signal from other places, or what does that look like? And then, as those get integrated in the database, we also have a traffic light system for trustworthiness of data. So red, yellow, green, and then it's mostly based on the source, but it's also based on, kind of how much evidence we have around that data point. And we use that to help us in our risk analysis around, our trustworthiness score.
Michelle really hit the nail in the head, and I'd like to build on that thought as well, more within the scope of what we are doing over here. So my recommendation is always keep it simple. Focus on technologies that you can understand, devices that you know. If you don't know the technology or device, well, make sure to get to know it, test it thoroughly. That way you will be able to interpret your results better, detect outliers, and contextualize your results. There's still a lot of overpromise on the market, and it's really our job to filter through all this noise and come up with the best recommendations. So in order to assure the best quality of the data, let's focus on what we trust. And if there is something new, let's understand it first.
Yeah, I think that's well said, both. I'd like to also add, you know, the upfront work, and I think that Jacek, you alluded to this, is really, really important, to make sure that you're setting yourself up well to obtain quality data. We kind of look at this in sort of a three-step process, which is, you know, what is the symptom you're looking at? How does that map to your signal? And then how does that map to your symptom? And so having these kind of three things that align with one another it's going to help you, say, choose which devices you may want to use for your study. I know that, you know, certain researchers we've seen across a variety of studies, you know, there may be a particular, you know, sensor that folks like to use, but it's really important to take this objective look and kind of map this all out before you even start designing your study. And then the second part of this is, you know, I liked what you said, Michelle, about having a human in the loop, but this is a little bit of a kind of a different take on that. We have to remember that our study participants are people. And when you ask people to do anything, there's going to be some unanticipated behavior there. We all like to think that even consumer wearables, which are designed in their own right to be user-friendly. I mean, I wear my Apple Watch every day. When you deploy these out at scale, you have to plan for every scenario and then contingency plan for when maybe your data is not meeting the mark. And so, you know, you've done your work up front to choose your sensors, but then you have to plan for how am I going to set my participants up for success? You have to make sure that you have validation loops in place. It's not set it and forget it and assume that the data is just going to flow in as you hope it will. So number one, making sure that you have some of these validation loops in place. Number two, that you're also helping your participants, you're training them. They have someone to reach out to, or you can reach out to them if you notice something that doesn't look quite right. And so it's really every step of the way. And you know, the planning work, again, I just can't emphasize that enough.
Abby, a follow-on question to that. Do you look at completeness scores across an individual's wearables data? That's something that we did when I was working with the All of Us program, just to get a feel for, you know, how complete was each individual's data. And I wonder if you do that and factor that in somehow.
Yeah, and that's a great question. And then that opens up this, you know, Pandora's box of like completeness and what does that mean? And setting that expectation of like, for wearables data, how much data are we expecting in a day? What is wear time? What, and then within that wear time, how much, you know, what is the threshold of data density required to perform those analytics? And so I think just, Michelle, like rolling all of those up to say, this is what we're looking for. It's not, as you alluded to, say a straight presence or absence of data, yes or no. It's like, are participants meeting wear time criteria? Within that, are we getting all of the streams that we expect to see? And really just having some of those flags. And what's great is a lot of this can actually be automated too. You can have some of these automated checks, you know, in the data. I hope that answers that question. I know we can talk about this for a really long time.
Absolutely. And we are starting to get some questions in the chat as well. So Michelle, I think the first one for you, and I know that you put something in the chat, but maybe sharing more broadly and expanding on it. I wonder if you can talk a little bit about how you're managing patient consent as it pertains to getting data from so many different sources, right? You even talked about media sources. So what does consent look like in that world of bringing together such heterogeneity within the data sets?
Yeah, so at the moment, we are not collecting individual level data, which would require that kind of consent. However, this is something that I think about in the future, as we try to look for additional data sources that can even get us closer to that infection point, such as the flu-smart algorithm that Evidation has developed. So I'm going to pass on that, but what I'm going to do is I'm going to answer a different question, which is because of the work that we do and the way that we collect data across multiple sources, from media, but also from, sources that are putting out data. Each of them have their own data licensing agreements. That it's not all just open source use as you will. A lot of them have specific requirements about what you're able to do with them, how you're able to share them, and so forth. And I think one of the challenges with doing this work more broadly is the fact that they're all different. And it would be really great if the industry could work towards some type of standard setting around, not only the consent process and how individuals are able to control who has access to their data, because that's an incredibly important point, but also how those data get shared at a population level or with partners in a way that makes it a lot easier to collect data across multiple sources and bring them together. So Abby, maybe that's a good question for you to follow up on.
Yeah, I think in terms of consent, at Evidation, we take a very privacy first approach. And so we operate on a model of what we call per use permissioning. And so every time an individual's data may be used in a research context, we ask that person, do you want to participate? And if yes, great. We can compensate you for the data that you share. And if no, well, that's okay. You can go back to interacting in the Evidation ecosystem. There may be another opportunity you want to participate in the future. We're actually about to launch a retrospective data sets. And we're really excited about this because we're going to be making available wearable data, PROs, other condition-specific types of experience surveys, symptom diaries, et cetera. And one of the things that I'm actually really proud of is that, yes, these data sets are available for researchers to do a lot of this preliminary work that we've been talking about here today. But as part of that process, the individuals have to actually consent every single time there's a new research use case. And again, we incentivize that participation because we believe that data is really inalienable from the individual, and so we incentivize that by compensating people innovation points when they choose to participate in a research project. So, Michelle, I think it's a great point. It's something that we take, very, very seriously. We would never share anyone's data, even for research purposes, without their express consent.
Excellent. And we did get another question in the chat as well, just around kind of the process of, do you start with benchtop testing before going in, within observational studies, before going into interventional studies, that type of thing. And Jacek, I think you put a response in the chat here, but I wonder if we can maybe spend a little bit of time thinking through what are the other, I don't know if it's data points, if it's processes, what evidence do we need to build in order to have trust in moving something from more of that observational setting into interventional studies that we see being run across the field for investigational products. So Jacek, do you mind expanding on that point a little bit?
Sure. So let's actually start not from the beginning, from the benchtop, but from the goal, right? What do you want out of your measurement? Let's say you want to make sure that it measures what it's supposed to measure with a good noise characteristics that allows you to see signals, right? So naturally we will think, okay, let's focus on the outcome measure in my population and go immediately to the study with subjects. They might be healthy or immediately go to the disease population. However, we are running into a risk of misinterpreting our results if we do not understand things like confounders in our measurements, if we don't understand things like in compliance in our measurements. So we were discussing the completeness of wearables data, right? If we don't know how to identify it, or if we don't know what are quantifiable effects of these confounders and otherwise in compliance, we might misinterpret our results. Many things, what happens is we think we see the signal, we think accelerometers measure calories, but when we take a closer look, all we measured was seasonality, because people move different in the winter and in the summer, right? So we need to be very careful designing the experiment and accounting for these confounders. And in order to do that, we need to understand basics of our measurements. And this is why I recommend, if you are agnostic to technology, thorough benchtop studies. Thorough benchtop studies identifying confounders, sources of noise, and then trying to project it to human behavior and designing the experiment accordingly. And the good news is, in many technologies, these studies have already been done. So you can do a lead review and only do a confirmatory small size benchtop just to make sure that you are comfortable with the technology. I hope that makes sense.
I think it does, right? Really understanding what are all of the potential variables that are in play, so that when we're using these data to form measurements, right, we actually understand what it is that we're measuring, and we're ensuring that what we're measuring actually does matter to the patients that we're all here to serve at the end of the day. So I think in that sense, it makes perfect sense. Now, noting we have close to 5 minutes left here today, I'd love to kind of do a bit of a round robin here. Some famous last words, right? Really understanding, we talked about several different case studies where we can leverage digital health data in different ways to improve clinical trial efficiency. What are, maybe like 1 minute each, what are action items we could take back to our organizations following this discussion that we've had in order to achieve operational efficiency at scale? So what are some of the takeaways? What should we go back and do differently than what we're doing today? I don't know who wants to be our lucky first volunteer here.
I'm going to be quick.
Okay.
Keep it simple. Keep it simple. Never overpromise. That way you're going to build trust and you're going to be successful.
All right, I can go next. I just want to +1 everything that Jacek just said. I will also add to that and say, I think collaboration is really going to be key, and bringing together respective expertise. I also think tokenization is really going to unlock a lot of opportunity. Maybe you're not an expert in something in the spirit of collaboration, but someone else might be. And so we've partnered with with DataVant. We're ramping up in the ecosystem to be able to enable our partners with some of the data that we have. So if we were to say keep it simple, also I would say be open to collaboration and leveraging respective expertise. You don't have to boil the ocean individually.
Awesome. And I will say, I just saw on LinkedIn a friend, John Wilbanks, posting that he was at a conference and there were 2 rooms next to each other, and 90% of the people were in one room talking about AI and 10% of the people were in the other room talking about data. And I would say that, without the data, we can't do the AI. And so thinking about the infrastructure for data is critically important. And, specific features around digital health technology devices can be considered around standardization of feature detection to help really drive that interoperability across. So I guess I would say, still focus on the data, don't just follow shiny objects around, and also, thinking about how we can standardize across the industry will help us to move faster.
I
And
love that.
don't follow shiny object should be the wisdom
Don't show it.
we remind ourselves all the time. People are doing that all the time. That's a mistake. Keep it simple. Keep being excited, right? But don't follow shiny object. Understand what you're doing so you can communicate well, so you can be successful.
Agree.
What a wonderful way to round us out here. Data is the foundation for all of the cool things that we could accomplish within this space. And certainly, I think building the right guardrails in order to ensure that we're actually able to make the most of that data, whether it's driving efficiency, having new scientific insights, et cetera, et cetera. Thank you, everyone, for joining us today. I do have a few calls to action in addition. Let's see if I can share the right slides this time. All right, so please definitely mark your calendars. Join us for our next webinar. We'll be hosting it next week, where we'll be launching our resources from our core measures of sleep project. So be sure to save the date. There's more information on our website that you can check out. It's going to be an excellent conversation. And the last thing before we take off today is we are ramping up for our next STEM project. The goal here is really, I think, focusing on how can we drive the adoption of digital measures as endpoints in clinical trials? What does the value framework for digital endpoints look like? How can we start to measure that? And how can we get better and better at actually deploying these endpoints? So again, tying back into that operational theme. So don't miss out. Be sure to scan the QR code if you are interested in joining us for that. And with that, I hope everyone enjoys the rest of your day, whether it's morning, evening, afternoon, anything in between. Looking forward to seeing you at our next time webinar. And thank you to our panelists for sharing so much wisdom today. We really do appreciate it.